Thank you, and it's great to be here. This is my first time to Australia. Beautiful country, beautiful city, and beautiful weather in Brisbane. Very impressed. So look forward to bring my family back in the near distant future. I'm a scientist, so I'm a scientist entrepreneur, even though I'm the CEO. So I still love science, love technologies. Maybe before my talk, I ask, how many of you have done RNA in situ hybridization? No? You have done one? Okay, good. How many of you have done immunohistochemistry or immunofluorescence then? Okay, quite a few more people. That's great. So just to introduce the company, Acd has been focused mainly on RNA in situ hybridization technology development and product commercialization. And what we provide are reagent kits that are both manual and fully automated kits for RNA in situ hybridization. Today, we have over 13,000 probes that are used by our customers worldwide. Covers about 150 different species, many viruses. Within Australia, we even have a probe for Tasmanian devil in one of the labs. And they are using this with success. And the beauty of this technology is that for any gene of interest, whether it's on the shelf or not, we can generate a probe within 2 weeks of time and with guaranteed success. It's been used by over 4,000 labs worldwide, and which have resulted over 800 peer-reviewed publications. It's been growing every new publication a day. The product has been produced under ISO 13485 condition, produced under GMP because the product is not only used for research application, but are now being used in diagnostic application in the US. And it's also being used in collaboration with pharmaceutical companies for companion diagnostic development to select patients for their drug. The company is acquired by Bio-Techne last year, so we're now a part of a larger organization where Bio-Techne is also the parent company for R&D systems, for Novus Biologicals, etc. So the RNA in situ hybridization technology basically is a parallel technology to immunohistochemistry that allow you to measure RNA transcript in situ, very much like immunohistochemistry measuring protein expression in situ. Over the last 40 years, a variety of different in situ hybridization technology has been come into play, but without much success because of lack of sensitivity, specificity, or workflow is also very complicated. And we have addressed all those problems with our RNAscope technology. And the essence of our technology is a special probe design that enables substantial improvement in signal-noise ratio to about at least 100-fold to the point of single molecule detection for any gene that you want to detect. And I will explain the principle in the later slides. And the result that you're getting is look like this. And you can tell, since you have done immunohistochemistry before, the chromogenic signal from RNAscope looks very much like immunohistochemistry. But this actually is a HER2 messenger RNA. This one is LGR5 for stem cells, EGF receptor, endothelial cell markers. At the fluorescent level, at the high resolution, it looks much like a fluorescent in situ hybridization result, where single dots represent single RNA molecules. In this particular experiment, the green dot is the BCR, the red dot is the ABL, and the yellow dot is the BCR-ABL fusion messenger RNAs. You can look at the expression in routine clinical sample, like formalin-fixed paraffin-embedded sample here, or you can look at the samples from blood, like PBMCs. In this case, it's a secondary tumor cell detection in the PBMCs. So since the commercialization of technology over the last 5 years, you can see a rapid publication by our customers. Now it's over one paper a day, with a major focus in the cancer space, followed by neuroscience, infectious disease, stem cells, HPV, etc. Majority, about 10% of the publication are in top 3 journals, Cell, Nature, and Science. So high impact research has been done by using this technology by researchers worldwide. So how does technology work? So assuming this is a target transcript that are within the cells, within the tissue environment that are fixed. And what we do is we design a pair of oligonucleotide probes, what we call double Z probe. And this pair of nucleotide probe binds to the target gene right next to each other. Each of the Z oligos about 40 to 50 base in length. About 25 base of it will bind to the target gene of interest. And there's a 14 base pair as a tail sequence. So when 2 of the oligo binds to next to each other, covering about 50 base target sequence, now you have a tail sequence of 14 plus 14. Now you have 28 base tail sequence that enables you to hybridize to a pre-amplification molecule, which then hybridize to an amplification molecule, which then hybridize to a label probe. And this label probe can be conjugated with HRP or AP, which give you chromogenic signal. Or it can be conjugated with fluorescent molecule, which can give you fluorescent signals. So with this design, a pair of double Z oligo binding to about 50 base of target sequence will be associated with as many as 400 of those label probes, which give you 400-fold of signal applications. And for detecting each gene of interest, we typically put as many as 20 pair of double Zs, covering roughly about 1 kb sequence. So you get this incredible 8,000-fold of signal implications for each target gene of interest. And we actually know for each transcript, we only need 3 pair of double Z, covering 150 base to give you single molecule detection sensitivities. So why do we build up such a big redundancies? For 2 reasons. One is for in situ detection of RNA transcript. Not the entire RNA transcript will be fully exposed to your probe. And another reason is that especially when you deal with routine clinical specimen like formalin fixed paraffin embedded sample, many cases you have a partial RNA degradation, so not the entire RNA transcript are available for probe hybridization. But that's okay, because in this particular design, if 15% of your probe bind to the target RNA of interest, you will generate enough signal to be seen at a single molecule level. So there's a tremendous amount of redundancy and robustness built in to this assay. And that's why this assay is robust not only for research application, but also for diagnostic applications. Now compare and contrast with a specific hybridization situation to target probe. You may have a chance of oligonucleotide that binds to a non-specific target. But in this case, you may have a single Z bind to a non-specific target, but chance of 2 independent oligo binds to a non-specific target and right next to each other are close to 0. And when single probe binds, the tail sequence is only 14 base, so you cannot hybridize to the amplification molecule. So that's how we achieve selective amplification of signal without amplifying the background, and therefore improve the signal-noise ratio by over hundreds-fold to the point of single molecule detection for any gene that you want to detect. So is it clear with the principle? Okay. And the workflow is very much like immunohistochemistry. For routine clinical specimen, basically you started with a deperiodization, then a target retrieval, antigen retrieval process, then protease digestion to allow probe penetration to hybridize to your target. So you then do target probe hybridization, followed with signal amplification step, and the label probe hybridization, which give you the signals. So the workflow very much like immunohistochemistry, which can be done in a manual assay for 6 to 8 hours with about 2 hour hands-on time. And because the workflow is similar to immunohistochemistry, it can be fully automated on the existing automation instrument for immunohistochemistry, such as the Ventana instrument and the Leica instrument out there. And with full automation, enable you to do translational research with the goal of eventually translate the results into diagnostics if the result proven to be of clinical utilities. So this slide basically proves that we have achieved a single molecule detection. Since the publication has been out for 5 years, I'm not going to get into the detail of the experiment that we proved the point. But you can see the individual green dots represent the HER2 messenger RNA, whereas the 18S is the red color because the 18S expressed in the millions of copies, so you don't see the individual dots anymore. Next slide. Even in the chromogenic assay, you can see those individual punctate dots. Again, each dot represent individual RNA transcript. So it's incredibly sensitive technology. And how do we know that this technology is more sensitive than traditional in situ hybridization? And one of the obvious experiments compare from this lab, where this part is the slides that are used in traditional chromogenic in situ hybridization using digital oxygen labeled probes. And this is the RNA scope results. You can see the difference is quite obvious. And under high magnification, you can actually see individual viral particles. So it's incredibly sensitive with this technology. How about specificity? With double Z design, you can truly enable incredibly specific detection of a gene of interest. And one of the examples of this is this particular experiment where you can use the human TGF-beta probe to probe either the human xenograft tissue, which gives you a signal, or the neighboring mouse tissue, which does not give you a signal. Or when you use a mouse probe, which will give you a signal on the mouse tissue, but not the human xenograft tissue, despite the fact these two genes are 90% in sequence identity. So it's very specific in addition to sensitivities. The technology also allows you to do the multiplex analysis. And as many of you know, that it's incredibly difficult to generate antibody to detect secreted proteins, cytokines, growth factors, chemokines, in this case, UPM, PI1, protease, and protease inhibitors. And with our assay, you can see highly concentrated expression in the stromal cell where the secreted protein are produced from. And in this particular case, they are only expressed in the stromal cell right next to the tumor, and they are not expressed further away from it. So in this case, the overall expression of these two genes are low, that even real-time PCR are difficult to measure them. Since each RNA transcript has a single dot, so you can quantify them by counting the dots. You can either quantify the expression through a manual method by just estimate the number of dots per cell, or you can use an image analysis tool to digitally quantify the expression at a single cell level. And there's a number of software out there available to measure the single cell expression. The one I showed is from Definium Biosystem. And there's also one from Indica Lab. And there's another one that's come out from Leica Biosystems. And we have continued to improve the technology, and our next generation in situ hybridization technology enable the ultimate level of sensitivity and specificity. So in contrast with RNA-scope technology, which we recommend a 20 pair of double Z to measure a gene of interest. Minimally, we require about 300 base of sequence to allow us to put 6 pair of double Z for detection. But with the base scope technology, we only need a single pair of double Z, 50 base of nucleotide, to enable single molecule detection of your RNA transcript of interest. And with this technology, now you can look at the splice junction detection. You can look at the mutation detection. And you can look at the very small transcript that can now be detected by RNA-scope technologies. And I'll give you one example of this, which is detecting of CMAT exon skipping, exon 14 skipping, where in this cell line and many patient samples where you see a new junction created between E13 and E15 exons. And we can design a probe across the junction of E13 and E15 and compare with the Y-type junction where the probe can be detected E14 and E15s. And you can see in the cell line of deletion cell lines, the E13, E15 probe will give you a signal, but not the probe against E14, E15. Whereas in the Y-type cell line, the signal was generated by the probe from E14, E15, but not the E13, E15 probe. As a control, the E12, E13 junction gives signal in both cell lines. So highly specific and sensitive detection of splice junctions. And same can be done for mutation detection. We have generated probe for a variety of different cancer, popular cancer-related mutations. And this is one of the examples where the Y-type probe will give signal in Y-type cell line as well as a heterozygous cell line, whereas the mutant probe will not give you a signal in the Y-type cell line, but does give you a signal on the heterozygous cell lines. And so this now provides a tool to enable you to see something that people have never seen before, which is at the single cell level within the tissue context, how the mutational clone come up and evolve during drug resistance. In a few months, you're likely to see a publication from a UK Cambridge lab that talk about how the drug resistance, resistant clone evolves over time, something that people cannot do before. So to summarize, what we have is a technology which is a universal RNA in situ hybridization platform, enables you to do molecular detection and visualize the expression within the cellular resolution and within the tissue context at the ultimate level of sensitivity and specificity to the point of single base resolution. It's quantitative because each of the transcript appears a single dot. You can count the dots to quantify the expression. And it's universal. It works for any gene that you want to detect. Many of the species, whether it's Tasmanian devil or seahorse or bat, a variety of different species, you can generate a probe for within 2 weeks. And it's quite a flexible platform because it can be chromogenic, can be fluorescent, can be singleplex, can be multiplex, can be manual, or can be automated. Very diverse application. I'm just going to highlight a few applications. One of the obvious applications is when you don't have antibody. And in human, from one of the Protein Atlas database, we know that up to about 70% of gene, you do not have a validated immunohistochemistry antibody available. And in those cases, you can certainly use RNA-scope technology as a surrogate to look at where the marker was expressed. And this is one of the examples of that, where the Wnt family of a gene are secreted protein. They are highly hydrophobic and they express at low level. So it's difficult to generate antibody for them despite the importance of the Wnt pathways. And this lab at Stanford have generated probe for every member of the family within 2 weeks. And you can see different members give you distinct pattern of expression in the hair follicles. And of course, I don't know the biology itself. It's just that it's beautiful to see the distinct pattern of expression by different members of this family. And same can be said about immuno-oncology. I know there's one researcher here look at the immuno-oncology where many of the markers are cytokines, chemokines, cytokine receptors are difficult to generate antibody to detect them in situ within the tumor microenvironment. With our technology, we have generated probe for every one of the members and validated them already. In this particular case, where there's a lot of splice variant, important disease marker in cancer as well, and they are only differ by a few bases. So in this case, not only you don't have antibody, even RNA-scope technology is not sensitive enough to discriminate them. And this is a case where the base scope technology can help you to detect and discriminate the different splice variant of these genes. Non-coding RNA is another class where now we have over 100 probes that are ordered by different customers. And in this case, there's no protein, so you have to use RNA-scope in situ technology to understand where they express to give you the hypothesis for potential biology and disease implications. One of the reasons people may use our technology is because the technology is more sensitive than immunohistochemistry. You may see results with immunohistochemistry, but you may miss something that you may want to look at the RNA expression as a surrogate. I just use a few examples to highlight that. And this is one of the examples, CMET, disease-important gene, a gene that have important diagnostic targets. In general, the expression of protein and RNA for CMET for different samples correlate quite well. But we have consistently found that at a low level, when immunohistochemistry is negative, you always see a few copies of a messenger RNA expression. And that is actually universal. And because when we look at the PD-L1, another important diagnostic marker for cancer immunotherapy, same thing, there's two diagnostic antibodies. They are negative for this particular patient, but you still see a few copies of messenger RNAs. And it's also true for HER2 as well. As you know, HER2 is one of the earliest target for personalized medicine drug. And a fraction of a patient highly express HER2 receptor in the breast cancer tissues. But there's a cardiotoxicity, and people do not know that actually HER2 is expressed in the cardio tissues, which can only be demonstrated by RNA expression. If you do immunohistochemistry, you don't see the expression in these tissues. Again, highlighting the sensitivity can give you indication not only about disease, but in this particular case, about potential toxicity that caused by the low-level expression of this gene. Another reason customers use this technology is to validate specificity of their assays. In fact, now many of the leading antibody-producing companies are using our technology to validate their antibody, especially when they're trying to generate a diagnostic antibody, which specificity is required. And I'll give you one of the examples that highlighting the need to validate the specificity of your antibody. c-MYC, of course, is very popular, important transcriptional factors. And the first immunohistochemistry antibody that comes out to detect c-MYC is 9E10. And over 100 publications have been produced using this antibody before the second antibody came about, the Y69. And one is against the N terminal of the sequence, and the other one is against the C terminal of the sequence. But if you look at this publication and look at the staining patterns, one stands at the bottom end of this tissue, whereas the other one stands the top one. So which data do you believe? In this publication, they used the RNA-scope technology to show that the newest antibody result correlate perfectly with RNA-scope. So all the previous work hundreds of publications are likely to be wasted giving you wrong information, just like this one. And in fact, over the last few years, there were at least half a dozen reviews, commentaries, publications highlighting the problem with antibody. And one of the review published in Nature says over $600 million was wasted on the antibody reagent alone each year. So that's a tremendous amount of waste, not only on the antibody reagent, but the researcher's time, the other reagents associated with it, so incredible waste. So when you have an important discovery by antibody, you do want to consider using orthogonal technology like this to validate your results. I mentioned the 2 diagnostic antibody for PD-L1. In this particular patient, the staining pattern is about the same, and they correlate with RNA scope. But in this particular patient, one gives you strong staining, the other one gives you weak stainings. So one would call positive, one would call negative. RNA scope is positive. So even though 2 diagnostic antibodies, all approved by FDA, can give you diverse results on the same patient samples. And if you look at this region, where both give you signals, but this is a region where RNA scope have no signal. If you look closely, these actually are necrosis tissue. The cell is already dead. So there's no RNA there, right? So just, this is why the specificity is something that this double Z design can impart the absolute specificity that you can feel confident about your results when you consider the use RNA scope technologies. Now, for people who do not do in situ detection of biomarker, who are only familiar with real-time PCR or microarray or next-gen sequencing, I want to make a point about the importance of cellular resolution and the tissue context, and additional biological insight that you can gain by look at the spatial context of the marker expressions. And one example is this particular one where previously the stem cells growth and differentiation was thought to be regulated by a paracrine mechanism where the Wnt family of genes and the protein are expressed in the neighboring cell that are received by the stem cell. But in this particular study, because they were able to co-localize the Wnt molecule within the stem cell, so now the new hypothesis is that those stem cell self-renewal are regulated by an autocrine loop, resulting to a publication in Science journals. So a biological insight that can only gain by look at the location of the expression of the ligand and the growth factors relates to the cell they are studies. Another example of that is the ligand receptor pair, where the ligand and the receptor are expressed in different cell types in a normal tissue. But in the head and neck cancer, there are 2 types of head and neck cancer patients. One type have overexpression of ligand and receptor, but they are still in different cell types. Whereas in another group of patients, the ligand and the receptor are expressed in the same cell type, so regulated in the autocrine fashion. And these 2 types of patients have distinct prognosis, and they also respond to drug differentially. So if you do real-time PCR, you will only see the overexpression of genes in both cell types, but you cannot distinguish these 2 populations that have different prognosis and respond to drug differentially. Again, localization information does give you additional insight about biology and disease process. Same is here in the neuroscience situation. 2 dopaminergic receptors in the same tissue, they are both expressed, but they are expressed in distinct neuronal types. Information you wouldn't be able to get if you do a grind-bind information method like real-time PCRs. So I'm going to just highlight a few additional research applications. One of the obvious research application is when many of them who do discovery research through next-gen sequencing or microarray or other methods identify candidate genes that involve in the disease or biological process. But how do you validate your candidate genes? Traditional people trying to look for antibody to see where they express and how they correlate with biology and disease. But now with our technology, you can rapidly translate those candidate genes and design probes and validate whether they indeed regularly overexpress or underexpress in your model systems without worry about whether the antibody is available or not. And this is one of the examples where the lab have used a single-cell next-gen sequencing to identify candidate genes that are implicated in early lung development. And once you get candidate genes, how do you know the result that you get by NGS is real? And they are using our technology to validate that indeed whether the markers they identified are expressed in the right cell type that they are looking for and the right level that they expected from the NGS results. The assay is very well suited for infectious disease analysis. In this particular case, I mentioned that the assay is so sensitive that you can detect individual viral particles. It also allows you to distinguish the cells that are in the active transcriptional state versus the cells that are in the latent state where they still have viral DNA, but there's no messenger RNA transcription, so they're not in the active state. Like in this particular case, where you can look at the RNA and the DNA in combination, identify those cells that are actively transcribed, and whereas other cells that only have the DNA location, but not in the active transcriptional disease-causing state. Something is very important in the HIV research when they're now trying to develop a drug to completely eliminate those HIV viruses that are hidden in the T4 cells, even though those patients appears to be free of viruses in their plasma. Zika virus, as you know, is relatively new, and the beauty of this technology is that once the sequence becomes available, within 2 weeks, you can generate a probe and be able to use that to understand the infectious mechanism of Zika viruses. So now we have over a dozen publications using our technology, and this is one of them from CDC. And because of this is a cutting edge, over half of those publications are in the news. So this is highlighting the speed of generating probe for your cutting edge research. Immuno-oncology is a space where this technology is ideally suited for because many of the markers you do not have the antibody, and also because that because of the complex the tumor microenvironment, that you do need to do multiplex analysis to identify what type of immune cell are present or infiltrated in the tumors and what kind of marker are expressed, whether they are expressed in the immune cell or in the tumor cell, and how do they correlate and interact together to regulate the cancer progression. So something that this technology can give you a unique insight on the disease process. Neuroscience is another field where traditionally in situ hybridization has been popular because many of the antibody doesn't work well in the brain tissue. With our technology, you now have the capability to not only look at the expression for many of the low copy expression genes, but also at a resolution that previously not possible. Now customers have used technology to look at the expression at the synapse in addition to cell body, achieving a subcellular resolution. And that was not possible with other technologies. In cancer, Another area is that you do next-gen sequencing, you may find some fusion gene, which often underlie the cancer. But how do you know that this fusion gene is the cause of cancer when you don't have antibody to detect the functional fusion proteins? In this case, you can detect the fusion messenger RNA as a surrogate for the expression of your fusion proteins. And this is one of the examples of that. I'm not getting into the technical details of it. I can often explain. You can even use the technology to look at the rare cell, in this particular case, a circulating tumor cell in a pool of blood. In this case, because the assay have such a high level of specificity, that you do not need to enrich the circulating tumor cell from blood anymore. You can see there's millions of the PBMCs where there's one micrometastasis of cancer cell. Only the tumor marker will only light up the tumor cell, but not any of the blood cell, whereas the blood cell marker will light up the blood cell, but not the tumor cell. So you can get this extraordinary level of specificities with this technology. You can also combine the immunohistochemistry with RNA-scope technology to look at both the RNA expression and the protein expression or protein markers. In many cases where cell type are defined by certain antibodies, so you can look at the gene expression and identify what type of cell that are responsible for the target expressions. And that's something that our customers have achieved success in many cases. So I want to use the remaining time to talk briefly about the ability to seamlessly translate your result from research to diagnostics, because the reagent kits that we use for research are the same reagent kit that we use for diagnostics. The instrument that you use for research is the same instrument that you use for diagnostics, only the software is different. So you have this seamless translation from research to diagnostics with our technology platforms. And this has been done over the last couple of years in collaboration with Leica Biosystems, where they are already one of the leading diagnostic labs in the world for immunohistochemistry. We also collaborated with them this is two public announced collaborations with biotech companies as well as pharmaceutical companies to develop companion diagnostic tests to select the patients for their drug under development. And so why would people use this technology for diagnostic application? Why this is actually an ideal technology platform for diagnostic application? For two major considerations. One is from analytical performance perspective, and that, like immunohistochemistry, it provides morphological context. It has a superior sensitivity and specificity, which I articulated earlier. It also requires very small amount of tissue that are routine clinical specimen. You can deal with a small tissue as small as fine needle aspirate or core needle biopsy that many of the technology may not have enough material for analysis. In practical terms, it's also an ideal platform as well because it fits into the current pathology workflow, where just like immunohistochemistry, you have routine clinical sample, you use existing instrument, and the result looks similar to immunohistochemistry, which can be interpreted by pathologists. In the US, there's an in situ hybridization reimbursement code, so you do not have to worry about reimbursement. And the speed of development for any target gene of interest is 2 weeks to generate probe, so enable you to very fast development of your diagnostic test. And there's already thousands of instruments worldwide for IHC and for this test. And speed is also important consideration because if you send a sample out to a reference lab to do a test, typically it takes about 2 weeks to get the result back. And with this technology, you can just run your tests in a routine pathology lab. So you can get a result the next day. So from both analytical performance perspective and from the practical perspective, this can be an ideal platform for diagnostic development in parallel to IHC and FISH. We already have a few tests in the market in the US in collaboration with Leica Biosystems. For example, this particular one, albumin, is a probe where you don't have antibody for because albumin is expressed very highly in the body. And it's useful for determine whether there's a liver metastasis. And the CMMV is one of the probe that is useful. I'm just highlighting here for specificity because this particular sample is positive for CMV but negative for EBV. And the next one, this patient sample is positive for EBV but negative for CMV. And one of the most popular probe is the HPV probe for head and neck cancer and cervical cancer. And the reason that this become emerging gold standard probe is because it's more sensitive than the current DNA-based test. We're more sensitive about 15 to 20%. And it's more specific than a surrogate test based on immunohistochemistry P16. Again, it's about 10% more specific than the test. So you have the better of both worlds of higher sensitivity and specificity. And today there's over 50 publications out there by a variety of labs worldwide highlighting its clinical performance and the correlation with clinical outcome data. And that's why this test it become an emerging gold standard, but now it's being used by almost all the leading cancer centers in the US. I'm not going to talk about the companion diagnostics because that's probably a little bit too far from this audience. Just to summarize, this is a universal platform for in situ hybridization of RNA, and it works for any gene that you want to detect in any species. Compared with immunohistochemistry, the advantage is the predictability, the sensitivity, and the specificity. And you don't have to worry about antibody performance anymore. And compared with the FISH, because it's more close to proteins, so it's more functionally relevant and more disease relevant, and they are likely to be used in more disease indications. So with that, thank you for your interest.